it's on now.
Okay, I think we can begin, and it's actually a great pleasure for me to introduce our very own Elihu Gerson, Director of the Maternal Research Institute in San Francisco, which is a non-profit institution devoted to social science research. And as many of you know, Elihu is a sociologist whose work is shaped by Chicago School of Sociology, American pragmatism, and has been working in sociology of knowledge, sociology of research, the way in which scientific teams organize their own work, and particularly evolutionary biology, computing, medicine. And the last big project that was actually based here that he was involved with was about the Museum of Vertebrate Zoology, Berkeley. And other people were involved in that project, who are locals like Elihu Gerson and Mary Sunderland. So let me just say also I'm very grateful to Eli for all the support and advice that he has been giving in the years to OHST first and the center now. So thank you. I understand he's now working on the study in the biochemistry lab in the Bay Area.
Mm-hmm.
But as far as the talk today, he started looking at the Hyena project in 2006 when he was invited by Professor Glickman to observe the meetings of his scientific team. And in fact, I'm very pleased to see that Professor Glickman is here today. And he's very kindly agreed to be taking questions at the end if there are. Professor Glickman is a cognitive neuroscientist who has been working with Donald Hebb, the founder of that very field, and has been at Berkeley since 1968. And affiliated with the Department of Integrative Biology and Psychology. And he embarked on the study of hyenas in 1983 with a focus on the sexual differentiations in hyenas. And these studies, among other things, has involved setting up and managing the largest captive groups of hyenas in the world. So I think this is a local story I've heard very much about, but never quite the full story, so I'm very eager to learn more right now from the direct people. So please join me in welcoming Elihu Gerson.
Okay, is the mic working okay? Great. Hold on 1 second, I have a small problem here. Does that work? Yes, okay. As Massimo said, I became very interested in this project in 2006 when Steve Glickman invited me to come look at a meeting of his collaborators. And I spent one entire Sunday in a room with 22 people, I think, going through 15 papers on the state of the research project. And I was quite blown away by the simple longevity of the project, which had been going on for over 25 years at that point. So I said, this I've got to study. And so I did. So here's the introduction to the talk, which neatly sets out the problem that we've got. This is a mother hyena, an adult. She's one of the crew that lives up in the hills in the colony here, which is unique. It's the only kind of living kind of the colony in the world. And that's her cub over there. And you'll notice that the cub is very happy to see us. And that sets up the problem for the research because that cub is female. But let me back up a minute. There are 4 species of hyenas. The striped, does this work? Should work. It's not working. The striped hyenas, the brown hyenas, the aardwolves, and the ones of most interest here, the spotted hyenas, who are the largest species in the group, and the only ones that have this rather peculiar genital anatomy that I'm going to talk about at great length. is a cutaway drawing made by one of the collaborators. Hyena females, the external, I stress not the internal, but the external genital anatomy of hyenas is peculiar in that it simulates the external anatomy of males. The internal anatomy is the standard sort of thing that you get in virtually every mammal, but on the outside it's kind of hard to tell them apart from the boys. The clitoris is large and erectile and very similar to a penis in appearance. There's no vagina. The labia are fused to form something that looks very much like a scrotum. And a very important point, everything, urination, fertilization, and birth all take place through that enlarged clitoris. You can see in the diagram the cub in the uterus and the bladder leading through the urethra. Giving birth through that canal is quite a process for the mother. It's very complicated. You can see that the birth canal takes a kind of sharp turn here, which makes problems because the fetus detaches from the placenta before it gets to that point, which means that any delay in that turn can make for pretty terrible problems. And in fact, the fetal death rate is extremely high. It runs upwards of 40%, especially for firstborns, because in order for the baby to be born first time, the tip of that thing has to tear to allow space to come out, so the room for the fetus to come out, which slows things down. So it's not easy being a mom. The hyenas are organized in clans, and the clans have dominance hierarchies. The females are much bigger, significantly bigger and more aggressive than the males, and dominate males in all respects. They typically weigh around 150 pounds or even more in the wild. Each clan has two separate hierarchies, one for females and their subadults, progeny, their cubs, and a separate one for the adult males. All the females and their cubs dominate all the males. Males leave the clan very shortly after puberty, go find another clan to move in with, and they have to work their way up the male dominance hierarchy before they can mate. Excuse me. Females mate as they will. One of the most important characteristics of the hyena social organization is competitive feeding. They hunt in ones and twos, but they're very noisy about it. So when they find something interesting, all the others come running, and they all gorge at the sight of the kill. They eat as fast as they can. It's a tremendous gobbling, stuffing process. They gorge and can eat a significant fraction of their own body weight at a single sitting. This encourages a great deal of aggression amongst them for jockeying for position and for maximum advantage, which means that those relatively low in the hierarchy wind up eating last. The adult females favor and defend their own cubs, so there's a fairly elaborate kind of organization grows out of that. The juveniles have their mother's rank at a kill, and as a result, the cubs of higher ranking females get considerably more food and protection than the lower ranking cubs do. Excuse me. Which has the effect of making maternal rank inheritable, so that the cubs of higher ranking females themselves have a disproportionately large chance of becoming higher ranking in their turn. So there's a kind of transmission of rank from generation to generation. Something which I haven't seen very much of or heard very much of in animal social organizations. So that's the sort of basic background biology to this. I'll come back to the biology in more detail in a minute. But for now, I want to talk about the hyena research network that has grown up around studying these problems. The problems that the female anatomy raises have been addressed. Oops, sorry. By a network of collaborators that's grown up over 30 years now. The work is centered on the colony that I've mentioned already. It's here at UC, tucked up in the hills. The field work that the network does is based at Michigan State University. I'll talk about that in a minute. Currently, more than a dozen senior scientists at 9 universities participate in this collaboration. This varies from time to time as this kind of steady recruitment of new collaborators as new kinds of specialties and new skills are needed. Others, drop off. But others, as well as this core group, others advise and participate in the process from time to time. Now, all of this got started. Lawrence Frank, in the late 70s, was conducting a field study in the Masai Mara Reserve in Kenya as part of his PhD thesis research here at Berkeley in the Museum of Vertebrate Zoology. So he was studying dominance and aggression amongst the hyenas in their social organization, and decided that he needed to be able to get measurements of hormone levels in order to do the study well, and that meant getting data on endocrinological material, and that meant also as well controlling the animals so that he decided he needed a captive population of animals, a colony. So he hooked up with Julian Davidson, who was an endocrinologist at Stanford, now unfortunately deceased, and they also in turn got hold of Steve Glickman, who were you in charge of the field station at the time? Steve was in charge of the field station up in the hills. The station had originally been founded by Frank Beach, a very famous psychologist who made very large contributions to the study of hormones and behavior. And they persuaded Steve, it didn't take very much to persuade Steve, that it would be a fascinating and very useful project to put together a colony of hyenas on the campus. And there's Beach becoming a collaborator with the hyenas, the cubs. This is Lawrence Frank collecting the first round of cubs that became the basis of the colony back in I guess it was 1982. With the help of the Maasai tribesmen that lived in the area. And this is, can you see this? This is the collection. You can see at least part of it, I guess. These are some of the first cubs that were collected. You can see a few of them over here. There are a couple more you can barely make out. Checking out the fence. These are, as you can see, quite small. They're very dark coloring. They haven't got their spots yet. They're born. They're almost black, and they get their lighter coloring and spots at what, a month or two? When do they get their spots?
4 to 5 months of age.
4 to 5 months. Okay, so these are very young, and they became the basis of the colony. Now the field observation. The collaborators, starting with Frank, have been observing the same group or clan of hyenas since the late 70s, 78 or 9, I think. Kay Holkamp, who you see here, took over the field study work in the early 90s. She worked with Frank in the 80s. And students from her lab conduct a variety of studies on a going basis. She typically has half a dozen students at a time, and they're essentially all required to go out there and live in the game preserve, studying the hyenas in the wild for at least some period of their training. And in recent years, in the last few years, these field studies have expanded to cover other sites as well as the original site, all still in the Masai Mara Game Preserve in Kenya. So, from the early beginnings, the network has grown to include a very large number of scientists, and more importantly, from my point of view, a very large number of different specialties. This list here is actually too conservative because there are multiple subspecialties within each of these fields that play a role, and I'll give you an example of that in a minute. Some of the collaborators bring specialized knowledge of specialized techniques, such as surgery. In particular, here in California, if you want to do surgery on a large mammal, you're obliged to have a license as a veterinarian. So the collaboration has typically had the services of a collaborating veterinarian throughout its history, but has also worked very closely with the animal care people here on the campus, which is another very kind of complicated collaboration. Other specialists add substantive knowledge from neurosciences, from developmental anatomy, from endocrinology, and so on. So here's an example of this. This is 5 people, all of whom are collaborators, important collaborators in the project, all of whom are mostly concerned with endocrinological work, but they all come from different specialties, and they all bring a different set of questions and a different perspective to bear on it. So this first gentleman on the left is Gene Wilson, who's from the University of Texas. He's an MD, who's mostly concerned, in principle at least, with human pediatric endocrinology, but has also done a very large number of studies, comparative studies on animals. Al Conley is a DVM, so he's been responsible for at least some of the surgery, but his principal concern is with comparative vertebrate endocrinology, and particularly with the early onset of hormone production in the fetal organs. So it's a very highly specialized topic. The gentleman in the middle is Melvin Grumbach, who is for many years chair of pediatric endocrinology at UCSF. Has made many important contributions to the field. Again, an MD focused on human development problems. This fellow is Paul Lick. I was hoping he would be here. He was for many years, he's a zoologist, PhD in zoology, and again, comparative endocrinology, but focusing primarily on reptiles, turtles, I think in particular. Was for many years chair of the zoology department here, and then dean of the biological service division, is now taking it easy and directing the botanical garden up on the hill. And finally, the last fellow with his back to us is Finn Cedery, PhD in organic chemistry, who became biochemist and an expert in hormone physiology. He unfortunately died just 2 years ago. But you can see how multiple specialties function, even super specialized variations in the same general specialty area have different kinds of skills and knowledge to offer the project. For most of the collaborators, excuse me, participation in this network is not separately funded. Their work, the cost of their work, get paid for out of their own budgets, and their contributions are supported by their own sources. So the effect then is that, they're doing this because they want to. Until recently, there have been very few formal arrangements amongst researchers. There are, recently at least, a small number of contracts between the universities who are insisting on various kinds of animal rights and intellectual property and other kinds of legal protections, which are not at all of serious concern to the scientists. The primary motivation for the collaborators, virtually every one of them, is that the topic of dealing with this problem is fun, it's interesting, it's frontier science, and most of all, it's fun. And I have heard that in one version or another, and some set of words or another, from every single one of the collaborators has volunteered this, right? It's just fun. So, a very important point tends to be understated. The full range of the collaboration spans all the biological phenomena from the molecular level to the ecological and macroevolutionary level. This is extremely rare and of very great interest to me. At the same, and I'm not aware of other projects that do this, right? At the same time, the work focuses on a particular suite of adaptations in a single species, which provides it with a kind of concentrated focus, which is again kind of hard to find in many collaborations. Now, and it's the size, the very unusual size and history of the collaboration, which makes this kind of combination possible. I have to stress that, excuse me, as collaborations go, this is very large, both in the sense of the number of people involved and the number of specialties involved, and it's been going on for a very long time. You just don't see this kind of elaborate kind of collaborative work very often. So let me change the topic now. Now that we've got a little bit of background on the collaboration, the central problem for the research is where does this extraordinary external anatomy for the females come from? What's its source? How do we explain it? And the starting point for thinking about that is the theory of mammalian sexual differentiation developed by Alfred Jost, a Belgian biologist in around the late 40s and early 50s. And his model is reasonably straightforward. In the case of males, in the upper part of the diagram, fertilized ovum contains the SRY gene, which in turn triggers the development of the male testes, the internal, excuse me, internal organs, which in turn generate hormones, male hormones, which in turn finally control the development of the external genitalia. So that's a fairly straightforward model. The comparable female model, there's no gene that generates the control, so the ovaries grow without being particularly triggered in any special way, and they in turn generate hormones. Although, as far as I know, Yost didn't talk about those hormones, did he? The hormones that generate the normal external genital anatomy of the females. Many people have taken this to perceived this to mean that the genital development in females is passive. Sorry. Which is not a good way of thinking about it. And I'll have a little bit more to say about that as I go on. Excuse me. This theory of Yost was extended to brain and behavior in 1959 by a group of psychologists. I'm not going to go into those details here, but the general assumption is that the brain, in the course of development, the brain is also triggered as, uniquely male or female in its structure. So the problem then posed by this model is where do the androgens that control the females come from? Because the hypothesis is that it's male hormones that are creating this bizarre anatomy. So clearly the hypothesis then is that androgens had to be circulating during the fetal life in female hyenas in order to generate this strange pattern. But then where would it come from? And by the way, in addition, those very same androgens might organize substrates or centers of aggression in the central nervous system that were subsequently activated by other androgens after the birth of the organism. So this has posed a major initial focus for the project. What the search for the naturally occurring, naturally circulating androgens in the female mammal, male hormones in a female mammal, and especially a fetal female mammal. Then the specific question is, can we talk about can androgens by the fetal ovaries, the first question, is it coming from the ovaries, all right? As they grow up, can they account for the penile clitoris and the pseudoscrotum of the female hyena? Well, here's some data. It's kind of a little bit hard to see, but I can work through it pretty quickly. This is a fetus at 33 days. It's normally 110 days. 110? Normal gestation period. This is so fairly early in the process. In fact, it's very early in the process. It's because it's before the emergence of the internal genitalia in the process. And they determined it was female using molecular methods. So we have a genital tubercle, and inside the genital tubercle, you can't really see it, but there's a little thin kind of channel which represents the emerging urethra inside the genital tubercle. That means that this fetus, which is female, is going to have the hypertrophic external anatomy and the scrotum, and you can still see, because it's very early, the Wolffian ducts, which normally will disappear in a female when the ovaries start to develop. So this is very early on. The ovaries aren't there yet, and nonetheless, we have the female, the typical female hyena anatomy coming into place. So that kills that hypothesis rather neatly. It's not coming from the ovaries because the ovaries weren't there. So next problem, where's it coming from then? Conceivably, it's coming from the maternal ovaries. This is a model put together by Fincideri and others back in the early 90s. So the idea is that I'm out of batteries. There's no justice. There we go. The mother's ovaries generate androstenedione, which is precursor to many hormones, both male and female. That the androstenedione goes to the placenta, where enzymes convert the androstenedione into both male and female hormones, in particular testosterone on this side and estrogen on the other. The structure of the hormones, you'll notice if you look, is very similar. All of them are very similar in basic structure. So conversions of one from one to the next in this precursor pattern are very easy. So a series of experiments to check out that idea were performed in the early 90s. Sorry. And things got complicated, but basically the basic answer to the question was very clear. Here's an untreated female. Sorry, I left out the critical point. In order to control that, the possibility that hormones were coming from the maternal ovaries, the pregnant mothers were injected with anti-androgen drugs that blocked the production of androgen hormones. And the results of that is, here's an untreated female with the normal development. Here's an anti-androgen male that came about as a result from that experiment. You can see they look very similar. And here's an untreated male again, it's kind of hard to tell the difference. So it's not the maternal androgens that are controlling this process. So, summary. The androgens in utero, in the uterus, account for the relatively small sex-specific characters of the males, but not for the basic structure of the genitalia. And again, androgens in utero account for the secondary characteristics of the female hyenas, but not the basic structure of the emerging clitoris scrotum complex. So this is an anti-androgen female. They don't have the androgen. The anti-androgens do have effect on treated animals. The length of the urogenital meatus, the length of the clitoris, and the width of the clitoral glands all are in a more feminine direction by comparison with other spotted hyenas, but the basic structure is not changed. What does happen is that the cubs who were treated in utero with these drugs found it much easier to give birth without running into the problems in the same way that other females normally do. 8 out of 8 cubs, treated cubs, were able to have first litter and their cubs survived, whereas only 9 out of 21 survived in the untreated group. And similarly for the first cubs, right? So what's happening here is that the blocking the male androgens for the developing fetus makes it a lot easier for a female hyena to have a cub. In some sense, she's less masculinized. So now the question arises: why go through this enormous cost? This is a mother here trying to deal with a stillborn cub. What justifies these enormous costs to the hyenas? Right? I'm kind of running behind, so I'm going to have to sacrifice that. So there's been a considerable amount, Steve, particularly a considerable amount of work on the. There we go. One very interesting thing came out of the field work. One of the Kate Holocamp students named Stephanie Delaniak developed a method for measuring androgen concentrations in scat, so that it became possible for the first time to get a measure of what the androgen level in a free-living animal was, and that showed that in this first diagram, the high androgen concentrations are associated with high rank. The lower numbers here are higher ranks. And that's a pretty significant, substantial correlation. When you look at their cubs and how often their cubs are aggressive toward one another, you can see that the high androgen concentration cubs are pretty aggressive, especially for the females. There's a pretty decent correlation coefficient there, but for the males, the white circles, it's not a very strong correlation at all. So there's clearly an impact on behavior. They're more aggressive, and this study that was done by Delaniak et al. Is the first time they had actual measurement of hyena level or androgen levels and aggressiveness. So, what's the bottom line? Was Yost wrong or was he right? Well, In part, he was wrong. The structure of the external genitalia is independent of the androgens in the gonads, and in the placenta. So he's just wrong. On the other hand, he's right in that sex differences in the external genitalia are dependent on the secretions of the fetal testes. So the model clearly needs significant refinement, but it wasn't completely wrong. So what's the top level message here? The development, differentiation development of female genitalia is an active process, not a passive process. It's simply mistaken to think of it that way. It's fairly clear by now, I haven't given you any of the details, but it's fairly clear that estrogens and other hormones are important in the developmental process, and there are still the possibilities of direct genetic effects which haven't been completely ruled out. Finally, there's more work to be done on talking about figuring out the exact mechanics of the placental metabolism. So there's a lot of research to be done before we understand in detail just what the development of this anatomical puzzle really looks like. Unfortunately, turning away from the biology now again, in 2007, very shortly after I started studying it, the group sent in a fairly routine proposal for renewing the funding of the hyena colony, the basic hotel bill of food and shelter and veterinary care, and that was rejected to everybody's irritation. Would set off a long period of efforts to raise enough funds to maintain the colony. Those efforts were partially successful. As it happens, the stimulus program came along in 2009, and that was a help. But despite these partial successes, the size of the colony has declined steadily from 30-something animals to what is it now? 13 animals today. And right now, it's not clear if the colony can continue past the next few months. So we're at something of a crisis point. The field research in Kenya will continue. It's quite thriving, in fact. But of course, loss of that colony will make it very difficult to answer many of the questions about development and evolution of the very unusual genitalia. So that program is in trouble. And there I'll stop because I think I'm just about out of time. And turn it over to you guys for questions.
Thank you.